closely following the lectures and have been giving the assignments, the exam basically will be from the slides, from the lectures. All I can say is just try to understand the concepts, and most of the questions you'll find is based on the concepts. Obviously, different institutes may have exams at different time, and you may have overlapping schedules. So this is really nothing else to be done there, but try to. Hope is you would have been regular with the lectures and the lecture slides, and that should be good enough to pass the course with good grades, not only just pass the course. Okay, Alka, we can move on to another question.
Yeah, so we can move to the question from discussion forum. So
Sure.
first question was, if a molecule show high alveolar deposition at 1 to 3 micron range, however, for it to enter the system circulation, it has to be below 100 nanometer, which is more likely to be exhaled with the airway streamline due to its low momentum. So how are the formulation prepared to get ideal alveolar deposition as well as systemic absorption?
Yeah, it's a tricky business. So I guess the question, just for everybody's understanding, is referring to the lectures. It's mostly looking at the lung delivery. So if you're looking at lung delivery through aerosolization or through dry powder delivery, so in that case, what happens is the best size to deposit in the lung is 1 to 5 micron in aerodynamic diameter, because there's obviously inertia, gravitation, Brownian motion, as I explained in the class. You will find that most of the particles in the 1 to 5 micron go deep in the lung. Anything larger deposits before deep lung, let's say in your bronchioles and trachea, and anything smaller tends to just get exhaled out and not deposit. Even if it goes to deep lung, it doesn't deposit and it just comes out. Now, so the tricky business is that if you want to deliver drugs to systemic circulation through lung delivery, then I don't think you want. It's easy to deliver the whole particle to the blood streams, and the reason for that is even these deep lung linings, they have very little pore size. So if you try to deliver a micron size particle, it will not diffuse. Let's say even if you're trying to deliver 100 nanometer particle, it will not diffuse through very easily, because these pore size in the lining of the alveolar regions and the blood vessels won't allow such big particles to translocate from the lung to the blood vessel. So really at that stage, what you are relying is that you make particles that deposit, and you're hoping that those particles will stay there for a while. And while these particles are staying there, they can slowly degrade. As they degrade, your drugs are released out. And these drugs are small molecules. They may have some partitioning happening across these barriers. And then you'll be able to get some pipeline of systemic delivery of the drugs as the particles are degrading. In terms of the whole particle moving from the alveolar space to the bloodstream, it's extremely difficult and will have very, very low efficiency. So I think, Alka, there's a question in the chat. Maybe we can take that first.
Yeah, sure. So the question in chat is, sir, why adjuvant and antigen both is required for innate immune response?
Yeah, so again, it's a question from our immune module. So Sagnik, actually, both is not required for innate immune response. Even one is enough. For example, if you just give adjuvant, that is going to cause very heavy activation of the innate immune response. Similarly, if you just give an antigen, there are immune cells, innate immune cells that are circulating, and they can detect that there's something foreign, something off, and they can take it up. So for the innate immune response, it's not necessary that you require both. But when you require both is for the adaptive immune response. So if you now want to get a very specific response, you would need both signals to be present. And the reason this is done is more maybe evolutionary, because maybe you don't, adaptive immune response takes up a lot of energy. You have heavy amplification of these cells that ends up happening in response to pathogens, in response to foreign entities. And so the body wants to be sure that there is a need to actually get to the adaptive immune response. And that's why maybe evolutionary, they have put in this condition that they should both be adjuvant as well as antigen. Otherwise, you will unnecessarily be activating your adaptive immune response. You'll unnecessarily get sick because all of your energy is now getting diverted to the immune system. And so likely that it's to prevent any frivolous activation of the adaptive immune response. I hope I answered that question. If not, you please feel free to ask another one, or feel free to unmute and speak about this. Alka, we can maybe move on to questions from the discussion forum again.
Yeah, so the other question in discussion forum was like, when an implant is introduced, according to the implant surface nature, the protein folding will change. Then that action might cause instability of the protein in the blood, eventually causes precipitation. So how does the protein maintain stability even after introducing a hydrophobic surface implant?
Yeah, so this is a question from our protein adsorption module. And yeah, you're right in saying that when an implant or a new surface is introduced, the surface will have its own hydrophobicity, will have its own hydrophilicity. And when you have proteins come in contact with that surface, they could rearrange themselves, they could refold, and maybe bring out their hydrophobic domains, or change the way that these things were initially folded. And absolutely, these action may cause instability in the protein. So a lot of time what will happen is a lot of these proteins will then get adsorbed on the surface that you have provided, or on which these proteins are basically interacting and changing the structure to. And generally these protein adsorption happen at a very while the adsorption individual forces are quite weak, there's many of them. So the interaction of a protein to the surface is very strong, and it's very difficult to take them apart. So generally speaking, most proteins that get denatured end up staying on the surface, and they don't start circulating in the blood and start interacting with other molecules to start causing precipitation issues. However, it is not unheard of. In some materials cause more denaturation of proteins. Some materials can cause heavy precipitation happening from coming in contact with proteins, and so that's where the biocompatibility gets defined, that you may say that this material is not compatible for this application because there is a lot of protein precipitation that is happening that may cause issues moving forward. So that's where I think one has to look at, and generally speaking, the materials that one has to choose has to avoid these kinds of issues. Okay, Alka, we can move on to the next question.
So next question is also from protein adsorption. So question is, does the protein undergo conformational changes when it is present in a different medium? If it is, so then protein molecules are stable in any kind of medium, but that cannot be possible. Please clarify this statement.
I see. Let me answer the first part first. So does the protein undergo conformational change in different medium? Absolutely, yes. Even just the change in ionic concentration, change in pH, change in temperature, change in presence of different type of ions, cations, and ions. These things can cause change in the protein structure. Then so that's the answer of the first part. In terms of the second part of the question, where it says that proteins are stable in any kind of medium, that's actually not true. Proteins are not stable in any kind of medium. It depends on what the medium is. Some proteins get denatured even in plain water. some proteins may require, some proteins may be more flexible in a sense that they may not get denatured in even low pH or high pH. So it depends on protein to protein, and it depends on what medium and how different it is from physiological medium that you are using that may determine whether protein is stable or not. Okay, I think there's another question in the chat. Alka, you can take that first.
So there is a question in chat box, like how FASL actually work, how it block T cell affected, fast cell the antibody.
Yeah, so Sagnik has asked this question. I don't know if we have covered FASL in this course. I actually don't know FASL very well, but these obviously there are receptors that seems to block T cell functions. There are some publications on it. I've been aware of this. So I am unfortunately unable to answer this question on how exactly the FASL works. This more goes into pharmacodynamics of or the biological interactions specific to this particular ligand and receptors. So yeah, so I would ask you to maybe read some papers on this. Okay, we can move on to the discussion forum. But in the meantime, maybe if others have questions, please write them or let us know. Raise your hand now, because I think we are down to our last question at this stage. So it'll be great to have more questions come in.
Can I ask last question?
Yes, please go ahead.
So hydrogel don't dissolve in water, but in the chemical properties, then why it is made up of hydrophilic structure?
Yeah, again, an interesting question. Yes, hydrogels are made up of extremely hydrophilic polymers, and so one can say that, of course, hydrogels are also hydrophilic. That is true. But when it comes to the dissolution in water, it's sort of slightly differently one should look at. See, when we talk about these polymers or monomers and talk about their hydrophilicity and hydrophobicity, these are still small molecules. Even though polymers are large, individual polymer chain maybe a few nanometers at max, as opposed to when you have millions and trillions of those polymer chains come together to form a hydrogel, then you're talking about a really large structure, right? So at that stage, these are macroscopic structures. Sometimes you can see them through your eye. You can see a hydrogel through an eye. You can see a jello through the eye. So while jello is extremely hydrophilic, if you put this in water, it'll take a lot of time to dissolve. And that's because they're held together by physical interactions, or they may be chemical interactions that may not even allow these chains to move apart. So as a result, you have this macroscopic structure that is liking water, so it absorbs a lot of water, but at the same time it can't really be dissolved because maybe the chains that are there are too big or entangled with each other that the water can't separate them. So that's the answer for why hydrogels don't dissolve, and they do dissolve. For example, physical hydrogels, if they're hydrophilic, can you leave them in water, and if there are enough forces to move apart these physical chains, they will move apart eventually, and you'll get individual chains separated out and no longer hydrogel being present there.
There is one more question in chat box.
Okay.
Sagnik want to ask what actually transfection means.
So transfection is a process of introducing nucleic acids, generally foreign nucleic acids, into a host cell, and in this case mostly eukaryotic cells, that may be required for a certain function. And we've discussed transfection in quite a lot of detail in this course, where sometimes we are looking at maybe a protein is not being produced because there's some genetic mutation, so you supply an external gene, so that now that you have transfected that cell with another copy of that gene, it can start producing that original protein, that unmodified protein, to perform its function. It can also be used for vaccines. I mean, we saw with COVID vaccines, mostly it was basically transfection of mRNA that was being done. So yeah, so that's the general term, transfection. Okay, I don't see any other question right now. Maybe we'll wait for a minute or two, and if you have a question, please do type it on or raise your hand, and we can take it at this moment. I hope all of you are enjoying the course and achieving your course objectives for which you might have signed up for this course. Okay, there does not seem to be any other questions. So in that case, thank you everyone for joining in, and I wish you good luck for your exam. And please do use these concepts if you can for whatever you're trying to do, whether it's more learning objectives or research. And I hope you all do very well in the exam. Thank you.
So shall I make end for everyone?
Yes, please.
Okay.